Yeah, so I'm visible to all of the audience.
Yes, sir, clearly.
Okay, so good morning and welcome to the presentation in our series of clinical research. This particular topic which actually I am today taking is actually clinical research planning, and I think it is very important thing. The reason is that the finally you will find the clinical research, if it is lastly becoming very successful and useful, it is only because of proper planning. If planning is poor, whatever amount of effort you put, actually it is not successful. So one has to really concentrate and invest a lot of time and requirement for planning, because there are many people they can do to you, but planning is very important, where you really need lot of input and understanding what to do and how to do like that. So clinical research planning is very important aspect of any clinical research, and it is actually a very long thing to. So what actually I am thinking that I will divide my entire lecture into two parts. Today I will take the part one of my lecture, and I will start with my slideshow here. So as I talked that actually that is the thing I wanted to take. My actually the topic is clinical research planning part one. Now, whenever you are planning anything, not clinical research, I'm telling, okay, you want to travel or you want to build a house or you want to conduct some program or something, there is a general process planning. So there are some steps which are there in process planning. What are these? Are the general steps, nothing to do with clinical research. Any actually thing you want to do, you have to plan it. So what is the planning process which is generally adopted? First, the idea, okay, you want to travel, whether you want to travel to hill station or you want to go to some actually national park to see the wild animal. So you should have an idea. Then you should analyze. You want to go to hill station, okay, which is the hill station which is very good. Then you have to also forecast that when you are going there, what is going to happen, temperature, these, that, how you will reach there. Then you develop your entire plan, when you want to start, when you want to go, where you are going to stay. Then after everything you have to implement, and then finally you have to evaluate. So this is the general process of planning for anything which you want to do. Now let us come to the research planning process, which is very important for us. So what is the planning process? First, actually, you have to actually, what is your perception and opportunity? You should know that you want to do a planning or research. So what are the opportunities which are existing to you? You should have that first. Then second very important thing is that once you identify opportunity, you see that the objective to gain the same thing. Third thing is that I told the planning, which is actually I am going to talk to you. You got objective. The planning premises is very important. Identify the alternative, because whenever you are doing clinical research, not necessary that what you are planning is going to happen. Some issues may come. Suppose that is there. Identify the alternative and be ready with that. Evaluate the alternative. You may be having 1, 2, 3, 4 alternatives. Which alternative is very feasible for you, very cheap, which you so evaluate them. Also then select choice alternative. Alternative number 1, if it is not there, number 2, number 3, like that. So you should have a choice. And then you have to look into the formulation of your supportive plan. So this is actually the entire planning process for any clinical research. Then last actually establish the sequence of activity. You have to see that how you are going to do, what will be the first, second, third, like that you have to make. So these are actually the 8 steps for planning process for any clinical research. Okay. Now let us see that what you mean by research. We are only talking clinical research. So what actually you mean by research? By just definition, research is a systematic collection, analysis, interpretation of data to answer a certain question. You have some question, you systematically collect the data, you analyze it, you do interpretation, and you look to solve a particular problem or particular question. One thing is very important, that it is very crucial to follow a cascade of scientific steps. And these scientific steps are very, very important in conducting clinical research. This is one of the very, very important component. One has to understand that you have to follow a particular cascade in a scientific step, which actually I am going to tell, to conduct a clinical research which is very successful. Now, this slide is very, very important because it is going to cover almost every step which actually I wanted to talk to you. That is the step of actually scientific planning. You start with the selection of your area of interest, which area you want, infection diseases, cardiology. So this is your first. Second thing, that in that particular area you find out a particular topic, which topic you want to do. Like cardiology, you want to do ischemic heart disease or heart failure. So second is the selection of the topic. Third, you have a crude research question. You cannot have a final research question on day one. Have a crude research question. So many things you have to look into that I would like to do. Once your crude research questions are ready, the most important thing is that go for literature review. And this is very important thing, because if you invest more time and more effort in literature review, you will be more successful in planning your research. So this is very important. Once you do the literature review, you analyze and you find an answer. You wanted to do a particular study, you have done a literature survey and you have already found that somebody had done. No point in doing that particular study, drop that. But you have done a literature review and you find that nobody had done a particular study which you are planning, there is no answer available in the literature, then what you have to do? Actually, you refine. It was a crude question. Now you refine, research more specifically what actually you want to do, what type of data you want to collect, what drug you want to do. So refine your research question. Once your question is refined, again go back and see the literature, what are the information is available as per that research problem which you are looking for that. Prepare your research hypothesis, prepare your goal, prepare your objective. Once these are ready, actually design your study. After designing your study, write a protocol and all document which is necessary to do a clinical research, submit to ethics committee and take a approval. So once you have designed and all protocol is ready, informed consent is ready, whatever document is required, submit, take a approval. And once you take a approval, now there are so many people are available for a particular thing, that is actually your study population. All they cannot actually participate in your clinical trial because you have got some inclusion criteria. You say that I will take only age between 18 to 60. There are so many people, those who are 80 years or younger. So this is your study population. From study population, actually you have to go for really study, where actually you want select your population exclusively for the study. So population and sampling is another very important thing. This depends on your inclusion and exclusion criteria and select the people for your research. There are so many variables depending on what actually is your area, what topic it is there. So many variables will be coming, blood pressure, heart rate, or some investigation you want to do. There may be so many confounding factors. Very easy example is that I want to work on hypertension. In hypertension, my confounding factor is there. Somebody is very much obese. Somebody smoker. So these will be the confounding factor and many bias will come. So once you have got the variable, check what are the confounding factor which will play a role and what are the possible bias which will come. After that, the next step is that find out your research tool. When patient comes or participant comes, what are the things actually you want to collect, what data you want to collect, what instrument you want to use, how data will be collected. So you prepare your research tool. After preparing your research tool, always do a pilot study because you are doing first time. There is no data available, no answer. So suppose you have to include 200 or 300 patients just to do a pilot study to identify, okay, I'm able to do, these are the problems coming, so you can improve that. So it is always better to do a small pilot study to find out the difficulty and then improve your protocol. Make a complete work plan based on your pilot study and go for your main study taking this sample. Collect your data, whatever the data you have prepared in case record form, collect that data. Go for data management. Once all data is completed, then most important thing is that interpretation, because the correct interpretation only will get your correct answer. So after data management, go for interpretation. And once interpretation is there, the final thing is that you got your report and you have won your this thing. So these steps are very, very important and one should be very careful in following that step for the research planning. So this slide is very important for you to understand and follow this path to come to this stage. Now, important thing is that we want to do a clinical research. We have to be careful about actually the selection of the research area. How? The selection of the research area, the broad area of research is based on following. Number one, actually researcher's specialty, whether he's a cardiologist or he's pulmonologist or infection disease. So depending on the specialty, the research that actually will be the area he will be working. So depending on his specialty, he will do. He's a rheumatologist, never he will work only on the spot related to that. Suppose he's an infectious disease person, he will work on infection. So specialty of researcher is first thing. Very, very important thing is that interest. If there is no interest, no point in doing any research. You should be interested in doing research. You must have very good scientific background, and this scientific background will come by habit of regular reading. You must have experience. That's the reason actually we are running this course. So you will have an experience of clinical research. Experience is very important to select a particular area of clinical research. Actual need of research in this area is that available source and funding. You should have a funding body. When you want to do research, you need so many things. Instrument is required. You have to do some actually investigation. So funding is also important. If there is no funding, whatever you are thinking, it is very difficult to do. Now, once actually you have thinking that I want to do a clinical research, the very important issue will come that how I will select a research topic. Now, what is the criteria for that? Priority of topic of research depends. So you may be having so many things you are thinking, but once you think of a particular priority, what actually you want to do. So this priority will depend on number one, the characteristics of the problem. Actually, what is the problem topic there for today? Actually, we have got the corona pandemic. That may be a really very important problem. So one can take this topic. Impact on health. Always take a topic where you think there is a big impact on health. For example, corona today. Not that only obesity, or there is some small thing which is happening, better need not to. So if greater the impact on health, your topic will be better. What is the magnitude? How many people are actually suffering with a particular problem where you want to work? Now, globally, corona is there, so it is a good topic. Seriousness. There are some very small, like very simple skin disease is there, which is not very serious. So there's no point in doing. But there may be some serious disease where so many people are dying, or we don't have a treatment. So this is another thing for selecting your topic. Preventability. You have to really think that a particular topic I am taking, can I prevent? For example, now best example, the day actually corona was appearing, people next day started working on vaccine, and now we are successful in getting vaccine. So preventability is also very important thing is that, and naturally curability. Can I develop a drug? I am developing vaccine to prevent, but people will develop. So can I use a drug, or can I do a study with a particular topic to cure a particular disease? What are the available intervention? Now coming to corona, what are actually availability today is there? What are the drugs are there? What are the proposed solution? What you want to do? Characterization of the proposed study will depend on feasibility. We cannot think of doing something which is not feasible. Number two, cost effectiveness. Something you want to do where you have to use large number of instruments, these are quite expensive. It will be very, very difficult. So calculating of the cost for doing a particular topic also is one of the reasons for your selection of the topic. If you have got the money, somebody is funding in crores, okay, you can go ahead, which is very, very less common. Applicability of the result, this is very, very important. Always kindly try to take a topic that after you complete your study and getting a solution, that solution should be actually useful for the population, not that developing some drug where only few people are suffering. Better to select a topic. So selection of a topic for a research is very, very important based on these factors. Now next is after topic, research question. Investigator must make sure that he has a particular research question. Without any research questions, you cannot plan any clinical research. The question should be very clear and very, very specific. Never consider anything which is non-specific in clinical research. Then that should reflect in objective of the study. You are actually writing an objective of the study. So this particular specific thing which you are telling must reflect into your objective. It has no answer by common sense. Whatever you are planning, by common sense, we cannot get out the result. So that is also very important thing you have to think. It has no clear answer in literature. I told you that literature survey is very important. And in literature survey, you should clearly see that there is no clear answer in a question which you are thinking. This is one of the very important things for selecting a question. Finding an answer to question will solve or at least help in solving the problem of the study. So this is very important. We are finding an answer. So selecting a correct question is very important for benefit for the medical fraternity. Now I tell you that for conducting or planning any clinical research, my thing is that one of the most important thing is conducting a literature review, which is very, very important. Here I will give one personal example, and at the last even I will show you some of the slide of that. When Corona start working on, I thought that can we do something for Corona problem? I was doing a lot of literature survey and I found that many of the literature is mentioning that there's a role of vitamin D as a immunomodulator, and high dose of vitamin D can be used in corona because they found that many of the patients, those developed severe corona infection and they died, in all of them vitamin D levels were very low. There are some study in literature has shown that when high dose of vitamin D was given to these corona patients, they improved. So based on all literature, we design a study that high dose vitamin D can be used in corona, and then we propose a study to ICMR. I will be telling you in the last part of my slide what actually we have done. So literature review gave me the idea that vitamin D can be useful in the management of corona infection. So this is most important, and I will take little more time and more number of slide to literature review because this is most important as far as my knowledge is concerned. What is actually literature review? A literature review is a survey of scientific article, books, medical journal, dissertation and other sources which are relevant to particular issue, area of research or theory, providing a description, summary and critical evaluation of each work. This is actually a simple definition of literature review. Now, a literature review may constitute an essential chapter of a thesis or of your dissertation, or it may be a self-contained review written on a subject. In either case, its purpose is what? What is the purpose of this? What actually we are looking into these article is, number one, place each work in context of its contribution of understanding subject under review. You want to do a particular thing, you got some idea. So the purpose is there whether in a particular context of what you are thinking it is fitting or not. Second thing, this will help you in describing the relationship of each work, the other or consideration. It will help you to identify new way for interpretation and shed light on the gap. You will definitely able to identify the gap in a previous research which doing a research review. So this is another helpful. It will resolve the conflict among the seemingly contradictory previous report. You might have come across also for the COVID, people have mentioned that X drug is working very well. Other literature people published in journals, they are telling no, this drug is not actually. You might have already heard lot of controversy about use and not usefulness of hydroxychloroquine, like that thing will happen. Identify area of the priority of research, and it will definitely prevent a duplication. If you are doing a proper research survey, you will feel that at least it will prevent you in making the same thing which somebody has already done. And one another thing is that it plays one's original work. When you complete and you send for publication, this will be your original work. It may be from your thesis or it may be for your dissertation. So just don't duplicate somebody else has done. You should have your own original work, and this is only possible actually once you have a very good literature survey. Now when you are coming to literature review process, it has got very, very important component. What are the important component? Number one is actually you have to list. Entire thing what actually you wanted to do, you have to list. For example, I telling that I wanted to do the use of high dose vitamin D in corona. I downloaded 170 references, you can imagine, and in my protocol I put 65. So you have to list all the information from the literature which is relevant to your topic. Search, 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 and that should continue. And you learn that after searching, you learn that what to do. For example, I was telling I want to use high dose vitamin D. People will not like, because most of the people, they take very small dose of vitamin D. I was telling high dose. So for that, I learned that there are papers, they have shown that even very high dose of vitamin D doesn't produce any side effect. So I learned that I can use it. Then after that, you write your entire thing and it will prepare your protocol, and clinical research planning will continue. Now, how you go review? Development of literature review require 4 stages. The first stage is that problem formulation, which topic or field is being examined and what are the component of issue. This is your first. You have to have a problem, you have to formulate that. I already mentioned about the literature, sir. Find the material which is relevant, patient, which type of patient you want to include. Data evaluation, determine in literature what data has been done, what type of work they have done. So this is the third thing. And last, you must analyze and interpret, and then discussion the finding and conclusion for pertinent literature. So these are actually the 4 important components you have to consider whenever you are doing a literature review. Next is that source. We are telling, okay, we want to do literature review, we have to do this thing, that thing is there. But actually, what are the sources? We should look for the literature which are really useful for me for planning a very good research. Is most important. Commonly we look into medical or scientific articles. These are good sources, especially for up-to-date information. This is very important. If you take journals and see that the latest last month what is actually published, you get the latest information. They are frequently used in literature review because you get so many references and all information is up-to-date format. So this is one of the very important source for the literature survey. Second is, depending on the publication, these material may be referred as a material for your, because you have to put all these into your references. Now, sometime actually we also refer what is known as a non-referred journal. Some are standard journal which are medical, but sometimes we use it. For example, non-referred journal says there's a trade journal. Somebody is having publication where they are publishing about the trade, or there are some scientific magazines. These actually are less vigorous and standard screening prior publication is not there. You can take it, but don't take granted that whatever is published here is really true. So you should be careful in referring non-referred material. Non-referred material may not be checked as intensively referred material, but many can still consider useful, although not for scientific literature. Never use for writing any scientific literature giving a reference of non-reference material for a research. But for some idea, for getting some information or including something, yes, you can use it, but never put value as a scientific thing. Coming to the source, books are very good source. Remember that books tend to be less up to date because recently published, but when we refer, it was published almost 5 years back. So lot of old information is available. So that is another disadvantage. And it takes longer for a book to be published than a journal. It takes 4 years, 5 years, but journals are published every 15 days or 1 month or 2. So book is no doubt very good, but this is the disadvantage. They are still likely to be useful for including your literature review, as they offer good starting point. A person who is just starting a clinical trial or clinical research, yes, referring a book is very good. So those who are starting a good starting point, actually they can take book and they can have some additional up-to-date data. Then we get lot of conference proceedings. These can be useful in providing latest research that has not been, because it has been published. You attend a conference and you get a proceeding. Their literatures are there are references. They are also helpful in providing information about the people in different research area. You wanted to do a particular area. If you go and attend a conference, you can find out a particular person also published same work. So you can contact him and again you can track all his information and you can have a discussion. This is one of the very good advantage that the conference proceedings will provide you the information, who are the people in the world they are working, and you can write to them and you can understand and you can discuss with them. So the source of literature from conference proceeding is also very, very good thing to do that. Sometimes actually we get information and publication from government. For example, today very frequently we get the report issued by ICMR guidelines for COVID. So this is also very useful because they select so many references and they come with the guideline. So government and corporate reports also are very, very important source for the literature. Many government department and corporation commission carry out research themselves and they publish. Their published finding can provide a useful source for information depending on your field of study. So depending, suppose you want to do epidemiological study and you want to know that where actually this particular problem is more, then this publication is very, very important. Here I can give you an example. Somebody wants to do the study on pancreatic cancer. Now you want to know that, okay, in which area in the country pancreatic cancer is very high. There may be a publication from the government source that the pancreatic cancer, like ICMR or other things, that pancreatic cancer distribution is highest in particular state. And that is the reason you can use it. So this information also sometimes is very, very important, especially when you are planning a epidemiological study, which is very, very important to look for the reports published by various government and non-government organizations. Theses and dissertations are really very good. These can be useful source for information. However, there is a disadvantage. What is the disadvantage of using thesis and dissertation as a source? They can be difficult to obtain. Since they are not published, they are kept only in the lock and key in the department, but are generally only available from the library. You have to go to the library, and if it is available, you can use. So that is the difficulty. The student who carry out the research may not be experienced. This is another thing. Thesis is written by the student. He is not having experience, so we don't know that whatever he is analyzed and he put is really very, very good. So this is another disadvantage of using thesis or dissertation as your literature source. Internet is really very fast, very good. For example, I was telling that when I wanted to design a study with vitamin D for COVID, within 10 days, actually I could get 170 references only via the internet source. But one has to bear in mind that anyone can post information on internet, so quality I'm not sure, and I'm not sure that that is reliable or not. Published data is more reliable rather than anything which is there in the internet, because anybody can publish anything which may be false. The information you find may be intended for general audience. Many things which is there in internet, it is mostly for general audience. It is not very scientific. So it is not suitable to include for your literature review, but general information may be useful. So no doubt it is very fast, give lot of information, but these are two very important disadvantage one has to keep in mind when you are using internet as a source of literature. Now, how actually you to consider for that? One is that what are the author's credentials? You have seen one publication. You have to definitely find out, okay, this author, really what is his credential? Really is a good author? What are the other publications? Which type of journal he is actually publishing? So publication is not very important, but it is published by whom? That is very, very important, because with primary historical material, case study, narrative, statistics, recent scientific, everything depends on the author credential. The people, those who are having a lot of credentials, their publications will be there in high index journal. So you can find that this is actually first thing. Second thing is the objectivity. Is the author's perceptive even handled produces? He might have done something, but he is produces in doing something, and he might have done lot of actually contrary data, which is considered he is rectified and he has quantitated that. So you should be careful for that. Then persuade, if the author is actually having a convincing, you have got that thesis, are you really see that the whatever author has given, are you convinced with that or not? It is very important for you to understand. Then very important thing is the value. Does the work ultimately contribute in significant way understanding the subject of my research? You wanted to do a research in particular area. You have done a research such way, find out one actually publication, it is very good, but you check whether it has got any value related to your research, or it is just a information, may be very good, but it may not be relevant to your research. So value of that particular research publication is also very important. So these are 4 points should be considered when actually you are looking for a particular topic. Now you have done the literature review, you have got everything. Now you have to write a literature, and writing literature is most important, very important. Many people, they do literature survey, they collect lot of data, but when it comes for writing, they are very poor. They may not able to identify what is relevant, what is not relevant. And once they write, then somebody read, then he find out this is a useless thing written. It is not clarifying the points here. References are not given properly and they are not up to date. So writing literature is again very important. There are 3 components for writing literature. One is that you have to write introduction. You have to develop the body of your literature, and third is the conclusion. So these are the 3 main headings which will be there in your writing of a literature. Now let us see one after the other. Now coming to introduction, what you will write? Define, identify general topic, issue or area concerned, thus providing a appropriate context, reviewing the literature. So that is first thing you have to identify. Second thing, point out overall trend in what has been published about the topic, what is actually published, what is the conflict. You may find that there may be some conflicting in theory which is in publication, put that. Definitely talk about the methodology, what method he has used, evidence, what is the evidence, what is the conclusion, and finally when you are reviewing that, see that what are the gaps. So these gaps you have to identify in writing your introduction, because that is the limitation which you are going to raise in your objective. Establish writer's reason, point of view, reviewing the literature, explain the criteria to use, analyze and comparing the literature. So these are important things which you have to consider when you are writing an introduction. Now coming to the body of a particular project. In a body, group research study, you have to mention review of theoretical article, whether case study or a common denominator, actually what type of approach they have gone, quantitative, conclusion of author, specific purpose, objective that you put into the body in much more detail, giving the reference. Then at the end, in the body, you just summarize the individual study. You might have seen so many studies. For example, I have collected 65 references. Each I have given 2 to 3 line summary of each study, what actually the author is summarized, and make a very comprehensive analysis. You mentioned that particular author is telling it is happening like that. However, the other author is telling this is not the way, it should be done like that. So you have to put lot of comparison between the author and the publication and make your analysis when you are actually writing the body. Then conclusion is very important because many people, they don't have much of time. So they wanted to know that your literature is properly done. They simply see your conclusion. So one should be very, very careful in writing conclusion, what it should contain. Summarize major contribution of significance study. You met 100 publications. You take 4 or 5 which are really very significant in contribution and the article of the body for knowledge under review, maintaining the focus established into the introduction. This is one. Evaluate the current state of art. What is actually today people are doing, what type of investigation they are doing, what are the gaps are there, that you should very clearly write in conclusion that these are the investigation has to be done today because particular investigation which is done previously is not useful. MRI CT scan has to be used for the diagnosis of lung problem in corona. Like that, you have to put into your conclusion and conclude by providing some insight into relationship between the central topic of your literature review and larger body of this. So writing conclusion into these things is another very important thing. Now, after literature survey, everybody looked at what is your rationale? What is your rationale for that? So what is your rationale for that is there? You are actually looking for the important issue related to that particular thing. And that what is your having that is very important. First, actually, the study rationale have an explanation of the fundamental reason for this research. You should be very clear that this is the fundamental reason due to that actually you are doing this research. This is most important for your rationale. Second thing is that you have to very clearly justify this work by literature survey that particular thing has not been done, but if it is done, it will be useful. So second thing, justification of the work. So these two things are very, very important in considering the rationale of your study. Few things that work for me when actually you are planning a clinical research and wanted to develop a protocol and conduct a research, submit for the funding, you have to consider some very, very important things. What are those things you consider, which normally I consider when we are preparing a research protocol? That learn, use effective research strategy. Searching is very effective. You have to really look into the journal which are actually talking about your subject. Keep a credible research journal. There I told you that so many are there. Already those who are very good credential, having a much usefulness you should use. Write about everything you read. Don't bother what actually will be there. Whatever you understand from that point, write everything initially. Don't write literature review since beginning. Don't write. Write summary today. After do that, write a summary today and read other literature review. Just check the literature review and check what is there. So these are the things you have to first look into that when actually you are planning for it. And finally, that once you got it in your mind, ask various questions that if I do this, how it is going to happen. If I do that, I'm going to get anything. So ask various questions in your mind and note down all those questions as your objectives. Now we are coming to another very important thing, is that everything is that the goal and objective are very important. How you proceed for that? Research goal and objective. The goal, sometimes we also say aim of the study, and objective must state that the very beginning of the study, since they will guide the investigator during the process of formulating research question and hypothesis. So the aim and objective of any study is very, very important, because that will guide every investigator, or actually the people, those who are looking into that. They will also help in prioritization of the process. Now, actually, when you are submitting for funding to any of the organization, they receive so many actually proposals. So they wanted to prioritize that, okay, whether this proposal we should give. So the prioritization of your proposal will be depending on aim and objective. If your proposal and aim objective are very good, you get a prioritization and you are getting first funding. If it is very poor, you will not get. So prioritization of the process is also very important for preparing your goal and objective. It should be good, it should not be very poor. They will be able to, reader and consumer of the work, judge whether the investigator has achieved these objectives, because we start with the aim and objective and complete. So at the end, what is available is report. So report, anybody can check, okay, he wanted to do this study and he wanted to get this information, and this is the data output. So only by aim and objective I can find out that at the end of the result whether you have achieved the objective or not. So this is very important for research goal and objective. Now coming to goal. What is the goal or aim? It actually describes the aim of the work in very broad terms. It is not very concise, it is very, very broad. So aim is very broad and it has to be made into objective. What is objective? These are more specific and relate directly to the research question. They are more divided into two types. Many people actually, they say that aim and objective are same. No, aim is very broad. They don't have any specific thing. Objective only are very specific because they are actually telling about your research question, and mostly they are divided into two. One is actually your primary objective. Primary objective means what exactly you want to do. I'll another give example. You wanted to test a new drug for diabetes. When you want to check a new drug for diabetes, my primary objective there, when I give a drug, blood glucose should come down and glycosylated hemoglobin, the normal value is 6.5, it should come down. That is my primary objective. But I may also be having secondary objective. By the way, when actually I am using my drug, no doubt I am checking blood glucose and also checking glycosylated hemoglobin, because that will tell my drug is useful in diabetes. But I also know that in diabetes, the person develops kidney problem, there is peripheral neuropathy, there are some other problems. So I will also evaluate what is happening to kidney function, what is happening to nerve conduction. This will become my secondary objective or some side effect. So there are always two types of objective. One is primary, which is most important, which is directly related to your actually question or aim. And then you do something additional because you are already collecting data as by the way. But it is not necessary that all secondary objectives should be analyzed, but some may come very, very useful. Now coming to research objective, what it should have? It should closely related to your research question. Always objective and your research question should be very near to understanding.
covering all aspects of problem. You have to define all the problem in that particular issue and cover all and everything. Very, very important thing, never talk anything which is non-specific. You should be very specific. For example, you want to do example of diabetes. You want to do mild, moderate or severe cases. What should be the glucose level when patient is entering into clinical trial? What is the drop of glycosylated hemoglobin you want to do? So you should be very specific. You are using antihypertensive drug. How much diastolic blood pressure fall you want to consider, or you want that decrease in blood pressure is enough? No, it should be specific. I will tell as per definition that my drug will be only effective when the diastolic blood pressure from baseline should come then 20 millimeter of mercury, then all it is. So it should be very specific. So this thing should be very specific. Order of logical. You may be doing so many parameters, 1, 2, 3, 4. So you should have a order of logical. What you will do first, what will be second, what is third investigation, what investigation you will do or you will try to even avoid that. State action in verb. That could be evaluated. What is the meaning? This sentence is very important, that whenever you are writing objective, always write objective in the form of verb. For example, to describe, to identify, to measure, to compare. So use these verbs rather than just something very vague. So whenever you are writing objective, describe. To identify the complication of renal complication, to measure the blood pressure in patient with hypertension, to compare drug A with drug B in pancreatic cancer. Like that you have to write your objective with the verb. And what are achievable, taking into consideration available source and time. You have to have what is your source, what is your time which is available. You want to do for 1 year, 2 years, 3 years, how much time you want to take and what you want to achieve. So these are actually important things you should consider in writing your research objective. And mutually exclusive, no repetition or overlapping. Never write objectives that two things you are writing which are overlapping to each other. Independently, each and everything should be independent, no repetition of objective when you are writing them. Then you should remember the word smart whenever you are writing objective. So you should be smart like me to write an objective. You should be very specific what you want to do. It should be measurable. Whenever actually you want to do some study, it should be measurable. Somebody wants to repeat the same thing. He will use the same drug and he should also be able to measure the same thing what you have measured, blood pressure, blood glucose level or anything. So it should be measurable by other person. Achievable. Don't do anything which we cannot achieve. Better not to do that one which you cannot achieve in a particular duration or particular problem. It should be relevant. So many things are there which is very important. And last is that you should be time bound. Whenever you are planning a clinical research, prepare that I will do the study for 6 months, 1 year or 2 years depending on the type. So you always remember SMART and work smartly for clinical research planning. Now further research objective, it should be properly formulated, it should be specific objective, will facilitate the development of research methodology, collection, analysis, interpretation and objective. All these things will be only possible perfectly if your research objective is good. If it is very poor methodology, anything will be done. Somebody will be actually recording the blood pressure in the morning, somebody will be recording in the evening, somebody will be recording with the mercury manometer, somebody will be recording. So methodology will be non-specific. somebody will be collecting data in the morning, analysis will be done. So research objective should be written in such a manner that all these things is possible and which is uniform and data can be utilized for interpretation and can be useful for the human being. Now we start with research hypothesis. What is that? The research hypothesis is the statement of a research question in a measurable form. For example, I am telling that I want to know what is the cause why in a particular state in the country pancreatic cancer is very high. Again, I will give example of the same here. Somebody mentioned that the pancreatic cancer is very high in Kerala. I don't know whether it is true or not. But what they told, that incidence of pancreatic cancer is very high because they consume the fish that is known as a tuna fish. In North, people are actually vegetarian. Incidence of pancreatic cancer is less. But the incidence of actually renal stone is very high in the northern people and other things because they consume lot of milk product which contains calcium. So incidence of a particular thing. So my hypothesis is there. I have to check how it is. So hypothesis is nothing, an idea which actually I am looking for that to confirm it. I really want to know, okay, the consuming tuna fish is more responsible for pancreatic cancer or more calcium is responsible for kidney stone, I don't know. But that is my hypothesis to go ahead. Research hypothesis will change. From hypothesis, I am going to research hypothesis to define a prediction, explanation of relationship between one or more independent variable. I will take and divide the people, those who are taking tuna fish, those who are not using tuna fish, and see that incidence of pancreatic cancer. I will do serum calcium and find if the serum calcium is high, whether there is a more kidney stone. So from the hypothesis, I have converted it into my research hypothesis and now going for a dependable variant to find out the outcome or condition of a disease. So from hypothesis, I went into my research hypothesis to confirm my objective. Hypothesis, in other words, translate the problem statement into precise prediction and expected outcome. I told you my hypothesis that pancreatic cancer is more because of use of tuna fish, and now I am analyzing that the people, those who are not using tuna fish, whether they have got lead pancreatic cancer. So something like that I will proceed into research hypothesis. It must be emphasized that for haphazard guess, never do any guess. By that you have to, you should have some evidence, but should reflect the depth of knowledge, imagination, experience of investigator. So you have to look, find out whatever you are doing and hypothesis, whether that is having any meaning or not, or just casually. Don't do anything casual guess for planning your hypothesis. Now there are two types of research. One is null hypothesis. Null hypothesis that I know that, okay, tuna fish produces pancreatic cancer. I will do the study to actually say, no, it doesn't produce. So I go with the positive finding, yes, tuna fish causes pancreatic cancer. More amount of taking calcium causes kidney, but I disprove that. That is actually null hypothesis. Other is alternative hypothesis. Alternative hypothesis there, yes. If really people are using more, they are developing it. So null hypothesis there, I go with particular finding which is actually reported, and I want to actually say no, it is not true, and I want to disprove. And if I disprove, then my objective is over. Now I will give some of the example. For example, you are going for an observational study in a clinical research, how you will plan. Here is the example. that this area is actually family medicine. Topic is communicable disease like hepatitis. So in family medicine, there is a actually very common problem which is there in the population is hepatitis. I want to do a observational study. What I want to, my goal is to contribute to reduce the hepatitis in India through studying the public perception about the disease. I want to know from the public that why they think that the hepatitis is very common in that area. Another example is that they are finding that particular type of problem is very common in particular disease. Just I collect the data from that population to identify why they think in that. I have got questionnaire, I'll prepare and I will collect. My object is to assess awareness, knowledge, attitude and general public toward hepatitis in India. For example, now they are telling that they found that hepatitis was very common in a particular area in the town where they are getting a water supply from X origin. That means I think water is responsible for causing more hepatitis, and then actually we will come with the observational study that if we are using a good quality of water, we can definitely control hepatitis. So this is an observational study, by that we can do. There are many examples that increase in obesity by using fast food. People those actually are using the fast food into various places, they are more obese. So some observational studies are there which can be very common. People those working in a night for very long time in software people, they have got different type of problems associated with the psychological condition like anxiety, depression. That is observational study. So observational study plays a very important role in identifying a particular problem, and then you can suggest the alternative. Now let us coming to another where interventional study, where you want to use a drug. Example is that cardiology. In cardiology area, my topic I want to do the study on ischemic heart disease or coronary heart disease. My topic is that can I have something of prevention of the coronary artery disease or ischemic artery disease. By my literature survey and by my knowledge, what I understood that to determine the LDL and find out occurrence of MI, because many people have published that the people those who are developing MI, the myocardial infarction or having more ischemic heart disease, they have got more LDL or cholesterol is very high. So I know that more cholesterol, more cardiovascular problem, that is the actually primary objective. What I will do, secondary objective is that I just wanted to see the side effect of lowering LDL. I will use a drug and I will decrease the LDL in patient. That will be my secondary objective, to see that I am using a drug to bring down LDL to prevent a cardiovascular disease, but I would like to see this is my secondary. So my research question will be there, does a hypocholesteremic agent like statin, if I use in a patient, whether it decreases risk of MI or not? This is my question. My research hypothesis, the risk of MI among the patient treated with hypocholesteremic agent like statin A is lower than the risk among the patient those who are not using. So I am comparing people those who are not using statin and those who are statin they are using. Then I will really come to know that really statin is really useful in reducing the risk of MRI, and that is a interventional study what you propose. Here I was just giving an example of my own study which I found in April 2020, where I was telling that I wanted to use actually high dose vitamin D3 and magnesium in the treatment of COVID infection. So it was a randomized clinical trial using vitamin D and magnesium. I have justified why I use magnesium, because it has shown in the literature that for the immunological activity of vitamin D, magnesium is essential. if there is low magnesium, vitamin D is not effective. So I have designed a trial, and these were the primary objective. I don't want to go here. I have to take the patient with COVID-19, give them high dose of vitamin D and magnesium. These are actually my methodology. I described in more detail how much vitamin D I should give. I should give in very high doses of vitamin D, and then I should give magnesium. So these are the dosage, and what actually I wanted to record here is standard treatment. These are the doses. I told high dose, that is actually 4 lakh. We never consider 4 lakh with milk because milk help in absorption. So I suggested high dose of vitamin D along with magnesium of 500 milligram. It should be used because in literature it has been shown that high dose is useful as a drug for the treatment of immunological disorder and even in some patients with COVID. So this was my basis of designing a protocol when I was submitting. What are the parameters I wanted to do? I know that actually the C-reactive protein is very important. I have to estimate vitamin D3, I have to estimate calcium, I have to estimate this thing and find out and how long my drug will be used. So this is the scheduled visit. Inclusion criteria, exclusion criteria, endpoint. What are the endpoint I wanted to do in my protocol? I have prescribed because I want to see that the individual should become negative for RT-PCR because RT-PCR positive only are the patient, those who are included. If my drug is effective, it should become negative after the treatment. There should be reduction in the CRP level. There should be reduction in the complication of and speed of recovery, and they will be staying in the hospital for very short time and they will become asymptomatic in the hospital. So these are my endpoint which I wanted to see if I use vitamin D, whether it is going to happen or not. Then these are the study procedure, endpoint criteria, synopsis, who has prepared. So all these things are actually is the planning of clinical research duration. This is the synopsis. Convert this synopsis into Bing protocol. We have submitted for the study to ICMR and Government of India. Approval got, and then there is another study we have proposed where we wanted to use actually vitamin D, vitamin K27 and magnesium in actually prevention of the people of healthcare professionals, doctors, nurses, attenders, those who are attending the COVID patient, they are highly actually this thing. So till vaccine is not there, they have to use mask. It has been shown that vitamin D, vitamin K2 and magnesium can help in improving actually immunity. So we have proposed another preventive study in a healthcare worker to use vitamin K, vitamin D and magnesium in COVID-19. These are introduction, study details. These are the primary objective. This was a 5-week study. These are the treatment guideline. Again, I want to tell here, we have suggested 60,000 international unit of vitamin D which should be used. That means 1.2 lakh on first day, a single dose, and then 60,000 once in a week. So we have described what should be done in synopsis, how it should be followed, what are the scheduled visit, and each scheduled visit what will be done. That also has been done in the research planning for the prevention study of vitamin D in COVID. So we have designed one study for intervention in COVID patient, other is for prevention of the people, those who are actually treating the patients like doctors and nurses, to see both actually we have given for ethics committee approval and funding and actually working on further on these projects. So this is the methodology we have described. These are inclusion criteria. These are exclusion criteria. These are the various endpoints. So thank you very much. And we will continue with the second part, and let us meet again. Bye-bye. I think you have enjoyed my talk, and if there is any question, I will be very happy to answer. Thank you very much for your patience listening.
Sir, we have a few questions asked in the live chat.
Okay. In slide, yeah. Okay, I will be happy to answer.
I'm just asking on their behalf. Is there any study shows that one particular disease or virus should restrict coronavirus?
As for my this thing is there, I am not aware. There are some report is there about some, but still personally I don't know that whether a particular virus, if it is there, it is can be able to reduce the coronavirus. I don't know.
Okay, sir. One more question.
Yeah.
To compare efficacy and safety of different doses of metformin in type 2 diabetes.
Yeah, I may say this is a general question is there because there is a standard treatment guideline for treating diabetic patient depending on their duration of diabetes and actually the level of the blood glucose level and the glycosylated hemoglobin. There is a standard guideline that you start with 500 milligram and observe them. If they are not responding, increase to 1000, go to the maximum dose of 2000 like that. There is a standard guideline, and then they say that you have to regularly follow and monitor their glucose level and glycosylated hemoglobin. If they are responding, you continue. If they are not responding, you have to add other anti-diabetic drugs. So there is a standard treatment guideline for management of diabetes, and that is regularly approved by the various associations globally. There is a global association on diabetes control. They provide regularly the standard treatment guideline, which drug to be used and how it should be used, what is the dose, duration, everything has been given. Same is for metformin also, for diabetes.
Okay, sir. Thanks for the explanation. One more question from the chart itself, sir. How to overcome the data discrepancy?
How to?
Overcome the data discrepancy.
Which discrepancy?
They didn't mention any. It's generally they asked, sir.
No, which discrepancy of the word I didn't understand.
Data discrepancy, how can
you are telling data? You are telling data?
Yes, sir.
Yeah, data discrepancy, what you are telling, for that one of the best thing is that last time when I was discussing, I was telling the frequent monitoring and checking what is known as checking the source document and actually the source data. When you do clinical research, there is one actually thing which monitor has to do. They have to check the source document and source data. Source data is actually which is entered into case record form, and source document is that which obtained. Again, I'll give example of diabetes. When I'm doing a study with the new anti-diabetic drug, the data actually which comes from the biochemistry department, they give me the report of blood glucose level for particular patient for particular date. During clinical study, the same is entered into case record form. I have to check whether particular data which is actually generated in particular lab is properly entered or not. That is one. Second thing which actually also I wanted to tell you, because this is very important, related to lab. When you are doing an investigation where you are using a particular lab, it is very, very important that that particular lab should be well validated and it should have what is known as NABL accreditation. If the NABL accreditation is not there, and from there if you are taking data, then data discrepancy will be very high as compared. Suppose the same study is done in Center 1 and Center 2.
Suppose center 1 is using their own actually lab from their hospital, whereas center B is using the data, all the investigation which is done in NABL accreditation. Definitely there will be a lot of data discrepancy and you cannot conclude. So there are some prerequisite. Monitoring is very important. Standardization and validation of all the method which you are using for recording any parameter has to be done to avoid the data variability what you are asking. So there are some guidelines to prevent data variability in clinical research. These are few which I gave example.
One more question from the same person, sir.
Yeah.
How to overcome data disruption due to lockdown in a clinical trial? Can we re-enroll the patients dropped out due to lockdown?
The one thing is that when you are doing the study, depending on the type of study, for example, you are telling lockdown, you wanted to do a study, for example, you want to do study hypertension or rheumatoid arthritis. Whenever you give a washout period, after some time, the same patient will be having same hypertension. So you can definitely include. But if the particular disease is there which is short lasting or there is a natural recovery in that particular disease, it is not possible to actually recruit the same patient. For example, you are using some drug or you are doing some studies where you wanted to check whether it is effective, but there is a natural course that disease will improve automatically. You cannot include. But if you are having a washout period after a particular time and the same basal point which actually happened during the lockdown, you are able to get after the lockdown is open, then you can definitely include. I gave example of hypertension, diabetes or rheumatoid arthritis. In these conditions, after lockdown, if you give a washout period, you will get the same patient condition which was there during lockdown and you can reintroduce the patient. This is only the criteria.
Right, sir. Thanks for the answer, sir. Sir, one more question on writing this article, sir.
Yeah.
What kind of journals, maybe the national level or an international level, what kind of journals we can consider for reviewing the literature? Some examples so that students can get
Here actually I can answer that it depends on the area what actually you are working. For example, if you are working on rheumatoid arthritis, actually there are national journals, those who are having a published by the Indian Society of Rheumatology. Suppose you are working on something, a general topic of medicine, then there is a actually journal which is published by ICMR or actually Indian Society of the Medicinal Physician. So like that, depending on which area you are, for example, infectious disease journal, which is published by one of the national organization, are very important if you want to study anything related to infectious diseases. So everything actually depends on your area of work. Hypertension if you are working, or if you are working on cardiology, then there are very good cardiology journals are there. I'm talking of national. Coming to international, there is no problem. There are list of journals, those having a very actually high ranking value. If you go and see, there are the ranking value given for each journal. Higher the ranking value, that journal is like Lancet, then you have got BMJ, then you have got NEJM, then you have got actually other journals are there. So they have got their actually publication ranking point where that you can select. But everything depends on your indication or area of work. Journal, if it is there, there are some journal. ICMR journal is very good for your review, which you will be able to do. But some actually industry also are supporting and publication you can take, but we are not sure because there is a lot of biasness in publication of those journals. So one should be very careful in selecting the journal. High ranking journal should only be taken for your references.
Thank you, sir. Thanks for the information. Uh, Diti, any more questions by the students?
Yeah, I just forwarded you.
Yeah.
1 second.
Okay. Sir, is it better to do clinical research in an known reason or to do research in an unknown reason? So I think they're asking for the indication, sir. Maybe
They're
based on the
asking,
indication.
they're asking
Uh,
for
Maybe doing research for a known indication is better or for an unknown indication.
they're asking to do some research in some disease or something like that.
Yes, sir. Yes, sir. For known
So
indication, doing research is better, or for an unknown indication, doing research is clinical research is better to carry out. Is that question, as per my assumption?
so I understood actually they want a topic for dissertation, isn't it?
Yes, sir.
Okay, yeah,
Before
I can
conducting the
propose,
research,
I can propose, you can actually give my email ID to them if they write, then there are few very good topic for dissertation which actually one can take and do the study. Maybe intervention study will be difficult because that is quite expensive and it should be funded, but non-intervention, very good observational studies are maybe quite useful and one can do that.
yes, sir. I have. I'm going to provide your mail ID to the
Yeah,
students. They can.
you can provide mail ID, let them write to me. Few of depending on their interest, I can suggest some dissertation or there are some studies actually which I'm also having and also proposing. They are interested. I can suggest them to do and make a data collection for me also.
Right, sir. Thank you so much, sir. Thank you so much for your wonderful time. And today we have learned many things, many more things about how to connect the literature reviews, how to choose, what are the sources to review the literature, what points to consider, and how can one start writing a literature, what are the key points to consider while writing and while, forming a very good research paper or dissertation or any article. Thank you so much for your wonderful explanation, sir. We hope to see you back with one more very good topic very soon. We'll be notifying to all our students and all our viewers on the coming topic. Thank you so much for your time, sir, and thanks to everyone who are watching here.
So I will actually try to complete my second part, which will be having a more the thing, and I will try to that, and if possible next week I'll complete my second part.
Right. Thank you so much, sir.
So yeah. Okay, so it's okay. I think I took more time, but I wanted to, because this is very important research planning is very important topic.
Yes, sir. Yes, sir. Yes.
One should understand it properly.
And we have all the recording available in YouTube. Those who
Yeah.
have missed some portion or wants to go through one more time through the important points, they can go and watch the recorded video in YouTube.
Right. Thank you very much. It will be useful for me also to forward to my students because they are quite interested to. They may not be able to attend because of their duties and everything, but always they ask. And I'm making my archive of all my presentations, so will be very good for me also. Thank you very much, Uma, for giving me the opportunity to talk to your people.
We are honored to have you here, sir. It's our pleasure. See you one more time, sir. Have a great day.
Right. Thank you very much, Uma.